Okay, so my name is John Stewart. I'm coming from the University of Florida, and let me just start by thanking the organizers for putting together this meeting. It's been an incredibly diverse range of topics. I've learned a lot. I'm going to say up front that I'm not going to give the talk that's in the book because most of that has been published, and I thought rather than do that, I'd sooner talk about some very recent work. So this is really literally hot off the presses here today. So my group has been working in biocatalysis now for many years. We think of that as good chemistry. And we've done a lot of functional group manipulations over the years. We've done carbonyl reductions and carbonyl oxidations and alkene reductions and so forth. But at the end of the day, the essence of organic synthesis is to generate molecular complexity by increasing the carbon-carbon bonds. And as we've heard a little bit in some of the previous talks, nature has a relatively restricted repertoire of carbon-carbon bond forming reactions available. Almost all of them follow a similar pattern where we have an anionic carbon species adding to an electron-deficient carbonyl, typically an aldehyde, sometimes a ketone, occasionally an ester. And so I've just shown here three very well-known examples, just to sort of bring you up to speed if you're not familiar with this area. Aldol reactions, as catalyzed by aldolases, were pioneered in the 1980s by George Whitesides and Chi-Wei Wang, and were really instrumental in moving the field of biocatalysis forward at that time. They are very broadly tolerant for the aldehyde component. One of the drawbacks, however, is that they tend to be very, very restricted with regard to the enolate partner. And so there are various kinds of these. They've been very important, particularly in the area of carbohydrate synthesis. The acylowin reaction involves a decarboxylation here to form what amounts to an anion on the carbonyl carbon, followed by an aldol condensation. It involves a thiamine pyrophosphate cofactor. This has been used industrially. It's relatively limited in scope, but for certain compounds is incredibly important. And the last sort of very well-known one was done by Professor Gringel in Austria and also by Professor Sprenger as well as others, doing the addition of cyanide to aldehydes. And again, this is very broadly tolerant on the aldehyde partner. That's a theme that I'm going to come back to. And by choosing the right enzyme, one can generally make either enantiomer here. So these are three really well-known CC bond-forming reactions. And because so much of the work has been done in this area, we decided this is not where we wanted to work. Instead, what I want to tell you is a story of two different types of enzymes. What they have in common is they require this cofactor called pyridoxal phosphate. I'll show you the structure of the cofactor in a moment, but what this cofactor enables is the formation of an anion on a carbon adjacent to a nitrogen. The presence of this carboxyl, while it is an electron withdrawing group, actually has no bearing on the reaction. Everything involves the nitrogen. In the first example I'm going to show you, the anion here is going to carry out a Claisen condensation with this thioester, generate a new bond right here, and that's going to be followed by a decarboxylation. That decarboxylation is important because the formation of this carbon dioxide drives this reaction very much to favor the products. So we have a built-in thermodynamic driving force for the carbon-carbon bond formation. This enzyme is called aminolevulinic acid synthase, or ALAS. Catalyzes the first committed step in heme biosynthesis. This is going to end up being the carbons of heme groups eventually down the road. Very little is known about the substrate tolerance of this enzyme. I'll show you a little bit about some other substrates. The other thing is that it requires this CoA thioester substrate, and I'll come back to why that's an issue in a couple of minutes. The other story I'm going to tell you is our very early work on a class of enzymes called threoninealdolases. As the name implies, these are responsible for the biosynthesis of threonine. So for example, if R is a methyl group, that would be threonine itself. And in a more general sense, what these do is produce chiral 1,2 amino alcohols here with good control of stereochemistry, particularly at the center bearing the carboxylic acid. A number of people have looked at these. These accept quite a broad range of aldehydes. That's not too much of a problem. On the other hand, they're rather restricted, at least so far, to glycine. In a few cases, they'll accept alanine or some other amino acid. That may be something we're going to be looking at down the road with protein engineering. The main drawbacks they have right now is the equilibrium constant here turns out to be close to about 1. And so if you want to favor the formation of this product, which is, of course, the one we usually want, then the typical approach is to use a big excess of a reactant. That's typically glycine. And so we're just using Le Chatelier's hammer to force this equilibrium to favor the right products. But of course, using glycine in excess complicates the product isolation. And if this is a more advanced intermediate, then of course we might not want to use this in high excess. So that's going to be something we're going to deal with. The other issue is the stereoselectivity. I said they control this stereocenter very well, but they often have a problem controlling this one, and that's related to the reversibility. And so that's another thing that we're going to have to address eventually by protein engineering. So let me tell you a little bit about our ALAS work first. And just to briefly step you through this mechanism, because it's important to what I'm going to say, this is all worked out by my collaborator, Professor Gloria Fajardo at the University of South Florida. This is the cofactor that enables the chemistry, and at the beginning of the sequence, it's parked as a Schiff's base with a lysine in the active site. Both substrates come into the active site, and there's a trading of the Schiff's base for a Schiff's base with the glycine, and the other substrate occupies its position within the active site. And that turns out to be important because in the absence of this, the deprotonation here to form the reactive anion is essentially non-existent. And so this pocket actually has to be occupied in order to form the anion. Once the anion forms, we have the Claisen condensation. That brings us to the intermediate right here, which is exactly the product, except we still have this carboxylic acid. Now, interestingly, it turns out the decarboxylation, while it generates negative charge on this carbon, that negative charge is not stabilized by the cofactor. It turns out that the bond orientations are not correct, and in fact, it's a typical enol type decarboxylation. The cofactor is not involved in that second step, but the decarboxylation then yields the product complex, and then exchange with the lysine brings us back to the catalytic site. So it's a fairly complicated mechanism. What's important to notice here, though, is that there are some possibilities for changing out the reactants. For example, one mutant that Professor Fajardo has isolated takes acetyl-CoA at this position. And so we don't actually need the whole group here. We don't know yet how much other freedom to operate we have here, what other changes we can make. The other thing that's interesting is the carboxylic acid, as I said, is not required. In fact, another mutant will deprotonate the methyl ester. And so I think protein engineering may ultimately deliver us the ability to work on a generic amine and not need to use the amino acid. Now, that's kind of far down the line, but that's where we're heading here. But like I said, the problem with this enzyme is that it requires CoA substrates. And for the most part, the fact that a gram of CoA will run you about $1,500 from Aldrich is a minor nuisance for most people who work with CoA requiring enzymes. But in our case, we want to do preparative chemistry. We need to be using gram quantities on a daily basis. And we're just not that rich. So what are some other possibilities? Well, the original method actually came out of baker's yeast. Step one is take 6 kilograms of yeast and dissolve it in 50 liters of water. And that didn't look very attractive. There was a very nice paper in the early 70s by the group of Shimizu, and out of a liter of culture you could get about a gram of CoA, and that looked really good. And if we could ever make it work, we would gladly use that method. But in our hands, even ordering the strain from 2 different strain banks, and they swore it was the strain in the paper, we could never get any significant CoA. So I don't know why it doesn't work in 2014, but we can't make it work. I'm happy to talk about it later. So instead, what I'm going to do is use a semi-synthesis. We're going to chemically synthesize part of the CoA, and we're going to build on some work that was pioneered by Eric Strauss back when he was a PhD student in Tig Begley's lab. And we're going to put this coenzyme A together by chemically synthesizing this piece. Of course, the business end is this SH here, but it's linked to this piece via beta-alanine. There is a biosynthetic route, of course, to this piece, but it's fairly complicated and the enzymes are not that stable. By contrast, the kinase that puts this initial phosphate on is quite stable and quite active. The subsequent enzyme that adds the AMP part to that also works very well. And the final enzyme that puts this last terminal phosphate on is also quite stable. And so we've gotten clones for all of these. We've generated the enzymes. That works very well. We purified them by his tagging. And all that remains then is just to make this, which is called pentathione. And pentathione is not a difficult target. It turns out the trick to this synthesis is this first step. And it's not so much making the amide as much as it is isolating the product, because of the water solubility of the product. Now, for reasons that'll be clear later, we're actually going to work with a disulfide dimer. We've actually gotten this to work to an okay yield. Some further optimization, it might work better. The good news, though, is there's no chromatography step here, and that's also important to us, to be able to do this at scale. So once we've got this piece, we deprotect the nitrogen, and just heating it in the presence of pantolactone, we can make our pentathione as the disulfide dimer. Now, the reason we like this disulfide dimer is it's a built-in protecting group for the reactive sulfur. What was not known was whether the enzymes that we're going to use for the subsequent phosphoryl transfer steps, while they are known to take a variety of analogs here, would they actually accept an entire additional CoA piece over here? That was not clear. We hoped it would be the case, though, because then we could just store this thing as the CoA dimer and clip it with a reducing agent as we need it. We were therefore quite happy to find out the very first enzyme, we call PAN-K, puts the phosphate on, and some analytical chemistry showed not only did it do it on one side, but it also did it over here as well. And you can also add the adenyl group by the next enzyme in the sequence. This also works at both ends. The last enzyme also works at both ends. So even though this thing has a whole big additional piece over here, all these enzymes are still working perfectly well. So this is actually the disulfide dimer of coenzyme A. If you add a little bit of reducing agent, then in fact you make CoA and it coelutes and it does all the things it should. So at this point, I think we've now got a scalable synthesis of CoA. There's no chromatography here. There's one quick filtration through silica, and we are now applying this now to putting together some actual chemical examples. In particular, trying different groups on here to expand the scope of this. And eventually, of course, this is going to lead us to protein engineering. The other half of my talk I wanted to discuss is a little bit about threoninealdolases. My student and I spent a couple of months last year putting together what we believe to be the comprehensive review of all examples of threoninealdolases applied to non-natural substrates. I think the table, when it was published, runs to about 10 or 12 printed pages. So there's a fairly large amount known about these. Well, what's interesting is virtually every example uses simply a wild-type enzyme isolated from somewhere. Very little protein engineering has been done to expand the scope and expand the substrate range of these things. And that's ultimately where we're going. We're starting that right now, as a matter of fact. But what I wanted to show you this morning is just some of our very recent results. We started by making a small library of just wild-type enzymes. We chose these 4 out of the ones that have been looked at. About a dozen have been looked at over the years. We chose these 4 because the wild-type enzymes seem to have the broadest substrate tolerance. In 2 cases, there are also crystal structures of these. We're going to fill in the rest of those crystal structures later. And they're also fairly stable enzymes. So some standard molecular biology yielded plasmids that encode these genes. We actually put these as his-tagged proteins here to facilitate the purification. This just shows the expression is actually quite good. This is for, I believe, the Aromonas jandii. The other 3 basically look about the same, and as you'd expect, the major band is the one we want. We can isolate this by his-tag chromatography. The crystallization studies are ongoing. We've got some quasi-crystals here. They're not certainly diffraction quality yet, but I'm pretty confident we'll have those things solved. So that's where we are with the enzyme work. I don't want to go through any more of that. But what I do want to end up with here are just some of our recent chemical studies where, just to kind of get our feet wet, we've profiled these with some new substrates. So these are some aldehydes down here that we've been looking at recently. The 2 that are circled are actually ones that were known in the literature. We included these just to check our results, make sure that our enzymes are performing the way they were supposed to. The short answer is yes, in both cases they do. These are all novel aldehydes. We use about a 5-fold molar excess of the glycine over the corresponding aldehyde, and this yields then the substituted threonine. We do look at these over a time course. As I said before, the reversibility sometimes leads to an erosion of diastereoselectivity. And so we try to stop these as soon as our conversion has reached the end. It turns out we spent a lot of time trying to fix the analytical, and eventually we wound up with something that is quite reliable. And that's where the results I'm going to show you here come from. So this is essentially the 1-carbon elongated analog of threonine. We can make this in effectively 100% DE after about 24 hours with one of those enzymes that I showed you on the previous page. We can elongate that chain. Turns out this actually goes even faster for a long chain. So despite the fact that these are called threoninealdolases, they can accept actually quite a bit longer chain here, and they in fact work a little bit better. And 3 of the 4 did this. We don't know at the moment what the absolute configurations are. I have a guess, but since I'm not sure, I just prefer to leave it off the slide. We do know the diastereomeric purity, but we have to assign the absolute structure. So I don't want to speculate at this point. We can also put in a substituted pyridine here. Again, our A jandii is our champion again. That's also a very fast reaction. And we can even put a furan in here. Turns out all 4 will take this. This is one where the diastereoselectivity, though, leaves something to be desired. And I think this is probably going to be a lot of what we're going to find as we scan through that table. And I think this is really where we're going to start to see the opportunities for protein engineering. We heard a beautiful story of Januvia this morning. And I'm hopeful that by applying some of those similar techniques, which we do in my group as well, we might be able to achieve at least some improvement in those things. So just as a quick summary, I've talked about our CoA scalable synthesis. As I say, it doesn't seem like a big problem until you contemplate using CoA-dependent enzymes to do organic synthesis. And in fact, we had another project we were going to pursue a few years ago, and it ultimately was abandoned because the CoA supply was too much of a problem. So I think not only is it going to enable us to do this chemistry that I'm talking about today, but I think in general now, we're going to make CoA requiring enzymes feasible for organic synthesis. And because so many enzymes in nature build carbon-carbon bonds by making claisen condensations with CoA thioester intermediates, I think this might actually have some use outside of our own group. We've done a little bit of initial work on the ALAS. As I said, there are mutants already known out of the Fajardo group that accept a modified substrate here. We're going to see how far we can push that. We also are going to start pushing changes here, ultimately, of course, to get into just a generic amine. That is the real goal here. And in terms of the threoninealdolases, we have similar hopes for those, particularly can we get away from glycine and just make threoninealdolases that use just a generic amine. And the fact that a transaminase that was ultimately an amino acid transaminase was endowed with this ability, I think gives me some pretty good hope that it's going to be possible here as well. So as I say, I apologize if you came to hear the flavin alkene reductase story. I'd be happy to talk about it later, but since that's published, I wanted to focus on this. And I just want to thank the people who did this work. The CoA thioester synthesis was done by a first-year graduate student, Luis Mutard. Sarah did the work on the threoninealdolases, and Trey Powell is a third-year graduate student. He's done the ALAS work. Thank my collaborator Gloria Fajara for all of her support and all of her clones in the ALAS field. My colleague Steve Bruner for all of the clones for the CoA biosynthesis enzymes. Thank these people for the support and you for your attention this morning. Happy to answer any questions.
We have any questions?
That was very interesting chemistry. I'm not really closely involved in this type of work, but the structures you had, like particularly on one of the last couple slides, what do you think the pKa of that ester is right there, which you show deprotonated? Because that molecule looks, because you have three positions that are deprotonated. Esters are usually not too acidic.
Yes, so I probably should have mentioned this for people who haven't looked at these before. It turns out the ester here is irrelevant to the deprotonation. The deprotonation actually is feasible because this can now be delocalized down, and ultimately what happens is when you push the electrons down, they wind up quenching the positive charge on that NH down here. And so it's actually the cofactor, it's this protonated nitrogen that ultimately becomes neutralized by that negative charge up there. And there's a whole series of resonance arrows you can draw. And so it turns out, you're right, the carboxyl, the ester is probably not very good at stabilizing that. But it turns out that's actually not what's stabilizing it in the first place. It's all due to the nitrogen.
And I wonder what the pKa of that hydrogen is before it's deprotonated. I wonder, is that known, do you think?
I do not know if the Fahar group has specifically identified the pKa of that. My guess is somebody has probably looked at that because this is actually the very first step in a transaminase. As we heard about a transaminase this morning, transaminases do exactly the same chemistry, and what happens is you make that shift space and they undergo a deprotonation. So probably somewhere in the transaminase literature, somebody has probably tried to measure that pKa, but off the top of my head, I don't know.
Thanks.
Anybody else? Okay, give them a big round of applause, ladies and gentlemen.